speaker, Dr. Kunliang Guan. Dr. Guan is currently the chair professor of molecular biology and cell biology at Westlake University. Dr. Guan received his bachelor's degree from Hangzhou University and later went to the US got his PhD degree from Purdue University. He then completed his postdoc training in Jack Dixon's lab, also at Purdue. In 1992, Dr. Guan established his own lab at the University of Michigan, where he rose to the rank of full professorship and was appointed the Harvard Christensen Professor of Life Sciences in 2003. In 2007, he joined the USASD, where he served as a distinguished professor of pharmacology until 2023. He then returned to China to join Westlake University. Dr. Guan has made landmark discoveries across multiple research areas. His group were among the first to clone MAC1 and 2, key components of the MAP kinase pathway. His group also made major contributions to the establishment of the mTOR signaling network. He is the second most cited investigator in the mTOR field. More recently, his research has been focusing on the Hippo pathway. His works on the regulation of the Hippo pathway and its role in mechanotransduction, stress response, and cancer biology had a major impact in the field. Dr. Guan is one of the most highly cited researchers in molecular biology and genetics. In recognition of his groundbreaking contributions, he has received numerous prestigious honors, including the MacArthur Fellowship, the ASBMB Young Investigator Award, Mark Brazil Award, and election as a Fellow of the American Association for the Advancement of Sciences. Dr. Guan is not only a true scientist, but also an amazing mentor. I had the privilege of working in his lab for a year, and his supportiveness has not only inspired me, but also many other young investigators in the field. So without further ado, please join me in welcoming Dr. Guan.
Well, I really appreciate Pengfei for the nice introduction. Always glad to see people who went through your lab. But actually, I cannot claim any credit for Pengfei's scientific success. But he was a postdoc in DJ's lab before joining our group. And I would like to thank Guoming for the invitation. I'm so happy to see many of the friends. I still call it older friends. Older friends, when we're friends for a long time, does not mean that we are old, right? So, but it's hard to be a friend for a long time when you're very young, unless maybe I met him when he was 3 years old. So, actually, that is true. Actually, I want to share you a story on that. The first time when Mei-Ling and we met, both of us were postdocs. He was a postdoc in Rick Hugenier's lab. I was a postdoc in Jack Dixon's lab. Both of us were working on the protein tyrosine phosphatase. We went to a festival meeting in Copper Mountain in Colorado. And Mei-Ling rented a car, and we drove to a casino. Casino, the gambling place. And we all test our luck. And I'm sure you know who has the best luck. Meaning, he won a lot of money, lots of coins, and we got a free lunch for that. So if you next time go to Korea casino, follow Mei-Ling. So what I'm going to share with you today is about give you a kind of a broader overview of the Hippo pathway and a couple of the recent studies. One of them actually is ongoing studies in the Hippo field. Now, one thing I really do want to say is a lot of people credit the discovery of Hippo pathway to G.J. Penn. That is wrong. That is very wrong. That is completely wrong. That said, DJ is one of the most prominent contributor to the Hippo the Hippo field. He's clearly the leader. I'm just want to set up the history. So the first gene identified in the Hippo pathway is called the kinase WATS, WTS. And this was initially identified by two studies. One is from Peter Bryan's lab that was working on Drosophila looking for mutation, which results in tissue overgrowth. And at the same time, exact same time, she who is Qi Tian, that's the first paper from his own lab. He was an assistant professor at Yale University. So Qi Tian and Peter Bryan identified the WATS. And more important, equally importantly, a few years later, Qi Tian identified the WATS homolog in mammalian cell. Now we call it the LATS, LATS, which is a large tumor suppressor. Qi Tian performed the genetics experiment. So through a series of genetic studies in Drosophila, now the key component were identified. That's about the time we started to working on the Hippo field. So we know at the time, mutation, for example, in WATS, this is a WATS phenotype, mutation in Hippo, this is the Hippo phenotype, results in extra tissue cell number growth. And downstream of that, DJ Penn identified Yorkie, which is a transcription co-activated function opposite to the upstream kinase. So the upstream kinase limit tissue growth, inhibit tissue growth. So they are tumor suppressor. And Yorkie promotes growth. There is a maybe oncoprotein, if you wish to call it. And I want to show you one of the phenotypes is over here. So this is the wild type omotidia unit in the eye. That's exact number of cells. Every omotidia unit have exact number of cells. In the WATS or Hippo mutant, you can see the interomotidia cells, cell number increase a lot. And this is due to two factors. Those cells supposed to die did not die. And there are some cell division, cell cycle should stop, did not stop. Extra cell division. So that mainly, immediately tell you the Hippo pathway affects the cell death and the cell division, which results in extra cells of the interomotidia cells. And the Yorkie phenotype overexpression results in cell growth. Now, the components of the Hippo pathway are highly conserved in the mammals. And so the Hippo homolog is called MST, which is a protein kinase. We have two of them. And it has a regular subunit, is sorbitol. And downstream of that is a WATS kinase, homolog is the LATS, and it also has a regular subunit. Downstream of that is the Yorkie transcription co-activated. In mammals is YAP, and there's two of them, is YAP and TAS. And downstream of that is the TAD transcription factor, which is bind DNA. So in the Drosophila is SD, in the mammal is TAD. There's a 4 T. And it controls a very broad range of biological and pathological functions. It's very broad. And one of the most striking phenotype, those initially reported by Penn's lab and Fernando Camargo, showing YAP can control liver size in mammal. It's very dramatic. If you inducibly express YAP, the liver size increase up to, fivefold, 500%. Now I should say that's kind of a little bit time sensitive. If you induce in a young animal, if you induce later, the liver goes bigger, but not that many-fold, not like a 5-fold. Amazingly, this regulation is reversible. If you take the YAP off, then the liver go back to its original size. So that's make a generalized concept is the Hippo pathway control organ size. At the same time, we also found that YAP activity is elevated in a significant percentage of human cancer. In this case, in the hepatocellular carcinoma, about maybe 40% of the HCC has elevated of YAP activity. So that establishes the Hippo pathway, control organ size, and also implicated in cancer. Hippo does actually more than that. So during development, the Hippo pathway is really important in development. So knockout of those genes are all embryonic lethal if we completely remove that. And one of the very first cell differentiation specification in mammals, you see the fertilized egg from a ball of the cell, the exterior cell, the outer cell, will differentiate to trophoblast, which forms the placenta, the extra embryonic tissue. And this is the YAP activity is necessary and sufficient to drive the trophoblast differentiation. In vitro, for example, if you use a human ES cell, if you artificially activate YAP, it will drive to a trophoblast lineage. The reason is the outside of the cell, it will become apparent why the outside cell is important. The outside cell has high YAP activity in the nucleus, which directly induce CDX2 expression. And the CDX2 is a trophoblast determinant transcription factor. This is beautiful work from Sasaki's lab. And this also tells us the position where the cell is very important. Now, in adult, it's not advancing. I dropped that. It should have nothing to do with. Oh, it does. Oh, I must have heard the mouse. Okay, so in adult, for example, this DJ's lab showed if you knock out the YAP in mouse in the intestine, if you delete that, you don't have much phenotype, once it's after development. But if you give the mouse damage, then the intestine tissue needs to regeneration, wound healing. And you can immediately tell what type of animal is able to survive, repair. The YAP knockout animal died very rapidly, and the reason is that the tissue regeneration is completely messed up. So the message I want to say is the Hippo pathway is also very important for tissue homeostasis, to maintain the tissue homeostasis, wound healing. And furthermore, the completion of the cancer, what is that called? Well, I blocked it. Anyway, then analyze like 10,000 cancers, look at the change, and the Hippo ATGC. It is ATGC, right? Oh, Cancer Atlas. Yeah. That also, that study unbiased analyzed almost 10,000 human cancers. We found about a significant percent, maybe about 10% of them, cancer has altered Hippo signaling. Of course, like a p53 mutation, RTK mutation would have been much higher, but that definitely basically establishes this pathway is important in cancer. Now, I want to come back to the very beginning when we look at so when we start to look at that, there's really nothing is known. What are the upstream signal? Actually, even the downstream transcription factor, TEAD, was not known. We contributed to the discovery of that. So we really want to ask is, how the Hippo pathway is regulated? What are the upstream signal? The very first signal we identified is the cell-cell contact. You can see, like this cell line, at the low density, YAP is nuclear. At the high density, YAP is cytoplasmic. And YAP is a transcription coactivated. So only the nuclear form is active. The cytoplasmic form is inactive. And this is, identifies the first signal, and this essentially now is a dogma. Almost every Hippo study paper use YAP localization or phosphorylation as the assay. The reason is this nuclear cytoplasmic shuttling is controlled mainly or primarily controlled by phosphorylation. So when Binzor in my lab, he figured out the signal. So it is, in a lot of part, is cell-cell contact. So there's many types of cell-cell interacting proteins, such as like adhesion, molecules. Or recently, the Korea, also a membrane protein, they form homophilic interaction. Means the same two cells express the same surface molecules. They're binding to each other, then tell the neighboring cells that you have a neighbor. Don't come to my territory. And this is very important that we know in cell contact inhibition. Actually, the Hippo pathway, if we artificially activate the YAP, it will override the cell contact inhibition. And mechanistically, when the cell contact, it activates the hippokinase cascade. When the kinase is activated, let's phosphorate the YAP on multiple sites. One of the sites is create a binding site for 1433. In humans, it's called serine residue 127. So that phosphorylation results into 1433 binding, which captures the protein in the cytoplasm. In addition to that, more phosphorylation will result in recruitment of E3 ubiquitin ligase and degradation, ubiquitin degradation. So therefore, caused to YAP cytoplasmic sequestration and turn off the transcription program. At the low density, the kinase activity is low, unphosphorylated YAP in the nucleus, as I said, bonded to the transcription factor. So the very first signal we discovered is cell contact. The second signal we discovered is hormones. So actually, the very first one is a lipid, lysophosphoric acid, LPA. This lipid is known to bind to G protein coupled receptor. And the GPCR, which goes through the low GTPase to inhibit the kinase cascade, which activates YAP, which is nicely showing over here. If you look at the YAP phosphorylation, so here's the YAP protein, here is the serum starved one, YAP is phosphorylated, and it is in the nucleus, it is in the cytoplasm. If we use serum or LPA, is able to induce very strong YAP dephosphorylation. This is a phospho antibody, this is a mobility shift. A very common assay we use is called a PhosTag. It's able to separate the phosphorylated form from dephosphorylated YAP. Very dramatic. Phosphorylated form, there's a multiple side, 5. The dephosphorylation will migrate here. You can tell the phosphorylation to dephosphorylation, this is not like 1% or 2%, it's essentially 100%. We can condition 100% YAP is phosphorylated to 100% dephosphorylated. So it's very robust and very fast. And we know LPA, we identify from the serum, is the main active. Initially, we observed the activity in serum. Then we identified, turns out is LPA. I don't want to go so. Actually, I do want to share your story to the students. Some of you may, I don't know. So first, we discovered that serum has activity to regulate the Hippo pathway. We were very excited, right? Because no other hormonal signal that. Then I have a postdoc come to my lab. I told him, I said, serum has activity. If you identify what is in serum, that will be a cell paper, and you get a faculty job. And the postdoc was really excited, working very hard, and tried to purify the activity. He worked for more than half a year, get nowhere, never able to purify it. The purification never more than 2-fold. And then, we put set aside. Then a couple of years later, then another postdoc came, his name called Yifashing. I told him the same thing. I said, Fashing, serum has activity. If you purify that, that's a cell paper, and you get a faculty. I did not tell him the previous person failed, And he did, and he tried to purify that, worked very hard, did not able to purify. Eventually, we discovered it is a lipid. How we discovered that? He was really frustrated in purification collection, and how some of the senior people over here probably have done protein purification, right? You measure how much protein you have, how much activity you have, and you divide the activity by the protein amount. And then one time, he was not able to enrich more than 2-fold, and one day he told me, he said, the BSA has activity, bovine serum albumin. I said, what? BSA has activity? And then I said, maybe we found in the Hippo pathway sensing protein. But BSA is the most abundant protein in the serum. Then he next did some experiment. He told me some BSA has activity, some BSA has no activity. He said, I tested 4 batches of BSA. Immediately compromised. Why we buy 4 BSA? 4 BSAs, that's costing me a lot of money, right? Why we have different BSA? And then when we look at that, it turns out so-called the fatty acid-free BSA has no activity. BSA has activity. And this guy binds to BSA very tightly. BSA, the serum albumin, the most abundant protein in our serum, is one of the functions bind the lipophilic proteins, small molecules delivered somewhere. And then everything explained, we cannot purify because we are purifying BSA, because they're never able to enrich. BSA constitutes about like 40%. Means the best purification fold you can achieve is 2.5-fold. I don't know whether you guys get it. But anyway, it was an interesting story. Maybe we should talk after, during coffee time. But it's here I really want to. So another unique part about the Hippo pathway regulations, it is regulated by mechanical signaling. And the mechanical signaling was first discovered by DuPont in Stefano Piccolo's lab. If you plate the cell on the different stiffness, the matrix at high stiffness or low stiffness, you can see at a high stiffness, YAP is nuclear. At a low stiffness, YAP is cytoplasmic. The cell is round up, so the blue is standing for DNA. So mechanical signal. Actually, about the same time when Zhao being in my lab, we also discovered the when cell attachment, detachment, which is also mechanical signal, affect that. Now here I want to tell some of the students, maybe some of the junior PIs, the student, sometimes you should not listen to your boss. This is a very painful story. The paper was published in 2011, you can tell. I moved to UCSD in 2007, and we are working on the Hippo pathway. And there's a postdoc called Ian Liang. He got his PhD from bioengineering department at the UCSD, joined my lab. It's the very first postdoc I recruited at UCSD. And one day, he come to tell me, he said, he changes the liquid flow on the culture medium. He said, affect YAP phosphorylation and localization. I was very simple-minded, or maybe narrow-minded biochemist. I said, what is the liquid flow with biology? I told Ian, I said, Ian, don't waste your time on that. Do something physiological relevant. And he's a mechanobiologist. That was also kind of interesting. Now I talk about those are important. I said, forget about it. Don't do that. Unfortunately, he listened to me. He did not do that until at the time Nature sent me a manuscript and I read the instruction. I said, wow, Jesus, there's so much mechanobiology there. So it is my ignorance. I think otherwise we should discover that the mechanical signaling. Yeah. And I think among all the research papers in the Hippo field, this paper probably is the most highly cited paper. Unfortunately, not from our lab. So one message I want to tell you is sometimes you should not listen to your mentor. Okay. So let me very briefly summarize. So we know the Hippo pathway contains upstream kinase. We know it is more than MST1 and 2. Original was MST1, 2. We and others identify the MAP4K family. There are several of them can directly phosphorylate activate LATs. LATs phosphorylate inhibit the YAP, regulatory transcription. And there's a very broad range of signaling I mentioned about cell contact, mechanical signal. Later we show the integrin involved in the mechanical as a stiffness signaling, GPCRs, as well as many of the stress signals, such as energy stress, osmotic stress, and heat shock. And one of the unique part, although it's still the concept evolving, so a lot of kinase cascade start with the receptor kinase, diamylase, like EGFR, or the kinase upstream is a G protein. Many of the MAP kinase family is a G protein like a RAS, bound to RAF activate. We see there's a Hippo pathway at the very top. It might be a group of a phosphatase complex called STRAPAC. STRAPAC stands for striating, interacting protein, phosphatase, and kinase. It's a phosphatase kinase complex. So one of the things that we think, for example, in response to serum, one of the mechanisms is, this is the phosphatase. This is the kinase. The kinase can form a dimer autophosphorylation activation. And if the phosphatase is not there, when you signal come, maybe somehow the phosphatase is recruited to the kinase and dephosphorylate and inactivate that. And a more elegant word recently from DJ's lab that's showing that's maybe so-called phase separation, also like a protein-protein aggregation, brings the kinase phosphatase together. So which I'm trying to propose the Hippo pathway at the very top maybe is the phosphatase complex, which receive upstream signaling to inactivate the kinase cascade. So currently we are really working a lot on this. And one example I'm showing here. Over here we use degradation, DTAG, to degrade the strip protein. Actually we delete strip 2 and use strip 1 is endogenous tag. It degrades that. Once we degrade the strip, you can see where the phospholates. You can see LATs goes up very rapidly. So this is the serum starvation, has basal. This is in the presence of serum. So it's a very rapid, dramatic response by the phosphatase. So we are degrading a phosphatase regularly subunit, not the, you cannot inhibit the phosphatase. Inhibitors have a too broad effect. So I want to share you a little bit about the drug development. So I should declare I'm a co-founder of Vivace Therapeutics, which is developing, try to target the Hippo pathway. I mentioned to you the Hippo pathway is altered in cancer, not very common, but some cancer. So if it's simple, when you call the Hippo pathway, the kinase module, which is over here, and a transcription module. Now, kinase is very easy to target. The problem is those are supposed to be tumor suppressor. So inhibitor kinase will not do any good for you to treat cancer. Now, here is the transcription module. We know inhibitor transcription is very difficult. Right? Luckily, there's a very important breakthrough, I think, in the field. So here's the complex structure, part of the TAD, kind of green bluish, and the pinkish, which is the YAP TAD structure. And in the TAD, it is modified by palmitoylation. Palmitoylation is a lipid, 16 carbon lipid. That fatty acid, not lipid, fatty acid. That modification is buried inside the TAD and is important for TAD function. And actually, when I saw that manuscript, I was, because previously we discovered TAD interact with YAP, and my collaborator Xie Hui, the lab solved the YAP TAD structure. So I was very surprised why they're able to see that from the bacterial, while when we means Xie Hui did that, did not see that. I called Xie Hui, I said, why because this modification is auto palmitoylation. TAD is an enzyme. This is transcription factor. If you give palmitoyl-CoA, purified TAD will auto palmitoylate itself. I called Xie Hui, I said, why we did not see that? Then an hour later, Xie Hui called me. He said he went back to look at the electron map, the density. He said there are electron density in their structure, but they have no idea what it is, and the student just simply ignore that and said that's a solvent. Turns out this is a palmitoyl-CoA modification. So one message I will tell the student is you should be always pay attention to the detail of your experimental data. We just ignore that, and we failed to discover that. This turns out, because this is a deep binding pocket, and you can develop drugs to inhibit the, this is completely messed up. That's not my problem, I can tell you that. That's a computer switch problem. So, develop an inhibitor to block palmitoylation. And so now there's several companies using that strategy to targeting the Hippo pathway. So now we can target a transcription factor. because that transcription factor has a palmitoylation and inhibits that. And one of the models uses the mesothelioma. Mesothelioma is very common. Oh, it's not very common. It is mainly caused by exposed to asbestos. This asbestos is 石棉，就是一个construction material. And in that case, NF2 mutation is pretty common, about 50% of that. NF2 is a key upstream component of the Hippo pathway. It's a kind of adaptive protein, which is recruiting actually NF2, based on Pengfei's lab, is recruiting the modulating the hippokinase activity. And when we mean the Tracy 10 tested, here the all 6 mesothelioma cancer cell lines, 3 of them has NF2 mutation, they're sensitive. These 3 cell lines has no NF2 mutation, they are not sensitive. And they are also able to inhibit the tumor growth in the mouse model. Now, NF2 mutation is very rare, but this drug approach maybe have a much broader implication is the following. So there's quite a few studies already showed, like if a cancer has an EGFR mutation, you give the EGFR inhibitor, inhibit the tumor growth, then a while later, the patient will develop resistance. And both in vitro, in cell culture model, as in clinical data, some of the cases, YAP activity come up. So high YAP activity can contribute to drug resistance. Now, this is a much broader market. And so in the mouse model, it's very clear. So here is the EGFR inhibitor alone has an effect, and will eventually develop resistance. The TAD inhibitor has no effect because this is not a TAD-dependent tumor. It's an EGFR mutation. But if you combination, it is very strong effect to prevent drug resistance. So this might be very exciting potential application. So if I list that, you can tell quite a few of the companies that are developing drugs in phase 1 and Vivace's phase 1-2 clinical trials. So of course we don't know the absolute data. The preliminary data actually is very promising. The TAD inhibitor can, so we select NF2 mutant mesothelioma patients as a first target. That's a problem. That's a kidney problem. It causes proteinuria, but it's reversible. It's manageable in the clinic. So I'm very excited. I'm really interested in hope it will work, because this is really so-called. We often emphasize innovation, first line of drug. This is conceptual new targeting the transcription factor. New compound, and we will try new pathways. Obviously we don't know whether it will eventually pan out or not. Okay. So now I want to share with you a couple recent studies. One is an unexpected role of YAP in small cell lung cancer, because generally YAP is elevated in cancer and is considered to be oncoprotein, because YAP promotes cell proliferation, inhibit apoptosis. And so let me introduce you, but we've seen something surprising. So small cell lung cancer. So lung cancer is pretty bad. Now, about 10 to 15% of the lung cancer are small cell lung cancer, because the cell very small. And the other 80% of them called non-small cell lung cancer. Pretty boring name. I think if the Drosophila guy working on that, I'm sure they will come up with some really interesting name. So, but the small cell lung cancer is the most aggressive in lung cancer, very bad prognosis. And the genetics is very simple. It's P53 and RB mutation. Essentially 100% P53, 80% RB mutation, probably 100% of RB pathway is inactivated. Or RB mutation about 80, because the other problem is this pathway. And the origin is different from non-small cell lung cancer. I came from neuron endocrine cell. The neuron kind property. What really caught our attention is, the very beginning is these results. So when we collaborating with Li Qingxie's lab from Fudan University, so my postdoc Wu Zhenming, so you can see when we do YAP staining, the small cell lung cancer has no YAP expression. I mean, no is not a 50% decrease. I'm talking about like a minimum tenfold. It's none. No, here we'll get a very few samples, but a lot more. The non-small cell lung cancer are there. And that is actually true if you look at the cancer cell line encyclopedia. So these are all lung cancer. The blue are SCLC, small cell lung cancer. And if you look at the YAP expression, they are silenced in lung cancer cell line as well. So, we were kind of surprised why YAP is so lowly expressed or silenced in small cell lung cancer, yet generally it functions as an oncoprotein. So first ask, what is the function of YAP? So we artificially express YAP in the small cell lung cancer. We also in vitro made a small cell lung cancer and can express YAP. And that expression level actually is not very high. It's comparable to normal, like a HEC293 cell. If the cell grows, whether you have YAP or not, no difference. We inject the cell to the lung, primary tumor has no difference. But when we look at the metastasis, for example, in the liver, it is very dramatically blocked the metastasis. So we express an oncoprotein which is suppressed metastasis. So metastasis is multiple step, right? First, the cancer cell need to leave the primary tumor, then need to get into the blood, then need to the distal side to colonize that. The first thing we look at is what about in circulating tumor cell in blood? Small cell lung cancer is very well known, among all cancers, it's one of the highest, has a so-called higher circulating tumor cell. People actually, small cell lung cancer, very difficult, normally don't do much surgery. One way people do is just get the blood from the patient, then isolate small cell lung cancer, then you can generate it like a tumor model. So when we look at the circulating cell, we did the following. The small cell lung cancer, we labeled with green. Another one, we labeled with the same cell. We labeled it with red, and it was inducible YAP. So we have a red cell, blue cell, the exact same, except one has inducible YAP. We mix equal amount of cells. We inject to the lung, and then look at the primary tumor, distal tumor, and the circulating tumor cells. So if you look at the primary tumor, both red cell, green tumor cell can grow. Okay? Now, when we induce that's under YAP induction, the distal tumor in the liver, only green cell, no red cell at all. Means the YAP expression truly block metastasis. And if we look at the circulating cell, without a YAP induction, we have a red cell, green cell, of course, a very, very low percentage. If we induce YAP, the red cell is no longer present in the circulation, the green cell is still there. So YAP block circulating tumor cell, and the YAP induction also extends the mouse survival. So we want to look at, so that means that the tumor cell is able to escape or enter the blood vessel. So we want to look at that. Now, small cell lung cancer, how many people have cultured small cell lung cancer? Nobody. It's very different from, they grow in suspension. They don't attach to that. Now, so under confinement, for example, like in tissue, means that you don't have a floating free in the liquid. You have a confinement. If you put some, solid up there, and so the small cell under confinement, this is smaller than the diameter. The cell form very elongated morphology. And if we label these cells, the green is the actin, GFP actin fusion in this small cell lung cancer, it migrated very fast. Amoeboid-like migration. It's very, very different from the mesenchymal migration. And the green which is the actin, is almost functioning like a motor at the end of the cell drive it goes through. It's a very fast migration. And we found when YAP is expressed, if we express YAP, the amoeboid morphology and the migration speed is very strongly inhibited. So YAP inhibit amoeboid cell migration with a result, presumably that's caused a reduction of circulated tumor cell and blocked metastasis. And YAP is a transcription coactivator, induce lots of gene expression, and we look at some of them, and we also guessed, and turns out that two of the bona fide YAP target, almost every, not every, many studies use these two genes as a target called the CCN1, CCN2. Those are secreted protein called the connective tissue growth factor, sometimes we call C. So these mediate the YAP effect. So here is I'm just showing the cell morphology. Under confinement, this kind of morphology, if you induce YAP, this morphology is blocked. And if we add purified recombinant CCN1, CCN2 protein, we see same effect as YAP induction. More importantly, if we knock out the CCN1, CCN2, then induced YAP is not able to block this phenotype. And here's some of the quantification. So two of the downstream targeted genes, CCN1, CCN2, mediate induced that amoeboid migration. So next thing we want to look at is, how YAP is repressed in the small cell lung cancer. And as I told you, small cell lung cancer has a very simple genetics, RBP53. So we look at, we put RB back in the wild type, that's a knockout, put RB back to the small cell lung cancer, also put a P53 back. And the results are actually put a P53 back, and most of the cells eventually died. RB put back, that grows slow a little bit, not a lot, but if we put RB, normally this cell has no RB, if we put RB back, endogenous YAP come back. So RB mutation contributed to the YAP silencing. And I should say, this phenotype is small cell lung cancer specific. Not all cells in knockout RB YAP goes down. Here is not our data. We just analyze the data. Non-small cell lung cancer, the vast majority has no RB mutation. Some of us RB mutation, YAP level is not changed. But in small cell lung cancer, majority of them has RB mutation. Here, many dots here. They have a low YAP. The wild type means the RB mutation wild type. RB wild type, YAP is high. So for some reason in small cell lung cancer, RB is required, not a small cell lung cancer. The PNAC, that's a precursor of a small cell lung cancer. You need RB to maintain its YAP expression and also affect the cell speed migration. So we want to look at what is the mechanism of RB. And we know RB bonds to many things, and the most famous are E2F family. And which we found is E2F, there's two groups. E2F like 1, 2, 3, maybe 4. E2F, those are direct bound RB to repress, to RB repress cell cycle entry. Then another group of E2F, like 5, 6, 7, 8, those are function as a repressor. That don't develop by RB, but nevertheless there are E2F family. So we found E2F 7 and 8 are very strongly downregulated in the RB rescued cell. And we look at the function of E2F 6, 7, 8, this is a subgroup in YAP expression. So this is a small cell lung cancer. We knock out E2F, this group. 7 alone is able to induce YAP. 7 plus 8 together give even stronger induction. So that means RB probably acts through E2F7 to regulate YAP, and that's indeed we showed over here. If we RB rescue the cell, YAP is come up. If we overexpress E2F7, we can block that. So now we know RB acts through E2F7. And E2F7, at the time we started, already known as a repressed gene expression. But surprisingly, nobody knows E2F7, 8, how they repress gene expression. So we look at how E2F7 repress gene expression. We did a protein purification, and very amazingly, the top group are the RQR, cause, well-known nuclear repressor gene expression. In the alcohol complex, include alcohol 1, alcohol 2, 3, when we purify with E2F7. E2F7 itself on the top. And they also include histone demethylase, histone deacetylase. So essentially, every one of those alcohol complex is bring down by the E2F7. And sure enough, if we knock out the alcohol 1, 2, 3, the triple knockout, you can see the endogenous YAP come up. And E2F7 knockout induces the YAP, decreases cell migration. Alcohol knockout also induces YAP, decreases cell migration. And then we want to, because there's a histone demethylase, deacetylase here, so we want to take, can we find a compound which induces YAP expression may able to block small cell lung cancer metastasis, right? So we look at that, and we tested a whole bunch of them, and one of them, antenal state. Sorry, it's moved a little bit. This is the antenal state. If we add this compound, induce YAP expression, is mRNA. Antenal state blocks the cell morphology under confinement, also block the cell migration. So antenal state, which is a histone deacetylase inhibitor, induce YAP, which is great. But if you pay attention to that, TSA is even a more potent, more broad histone deacetylase, does not induce YAP. Antenal state is a relatively narrow and less potent histone deacetylase. Right? If you see that data, your immediate conclusion is YAP induction by antenal state have nothing to do with histone deacetylase inhibition. Right? It must be something else. Actually, we try to figure out what is that something else. We get nowhere. And now we know why we get nowhere, because it is nowhere. So until we did this experiment, Zheng, I don't know why he did that experiment. This is a histone deacetylase inhibitor. This is a histone deacetylase inhibitor. He put both inhibitors together. TSA block the antennal state effect. That is really, really instrumental for us to figure out the mechanism. So I will not go to the detail. So the mechanism is the following. There's two different classes of histone deacetylase. The SYN1 complex, the alcohol complex. The SYN1 complex is positive for YAP gene expression. The alcohol complex media downstream of RB is negatively repressed YAP expression. And the reason is antennal state induced by antennal state is more specific inhibit alcohol complex. That's why induce YAP. TSA inhibit both of them, then blocks the antennal state effect. So great, now we have an antennal state able to induce YAP. So we want to look at it on the tumor metastasis. So first we treated the mouse with the antennal state. It is able to induce YAP very nicely. And when we look at the circulated tumor cells, if we put an antennal state, it's reduced very dramatically. And we also look at the, liver metastasis. It is able to block liver metastasis and able to extend the mouse survival. Now, all drug has non-specific effect, right? So how do we know the antennal state effect is due to the Hippo pathway, due to YAP? Luckily, we can very easily do the experiment. I told you YAP is not expressed in small cell lung cancer. Delete YAP, nothing happened to small cell lung cancer. If we delete YAP, the small cell lung cancer, the YAP deletion cells will grow tumor, will metastasize. Oh, now the key. When we treat it with the antennal state, the YAP knockout cell, antennal state is no longer able to block YAP metastasis. So that means we came up with the following model. So antennal state, by inhibiting the alcohol, this is not from our data previously, this goes through the YAP to result in, effect of YAP metastasis. So antennal state potentially could inhibit small cell lung cancer. Now, regarding clinical value, I think it's, if anything, probably close to none, 0. The reasons small cell lung cancer is very metastatic. Most of the patient as a diagnosis is already metastasized. Entinostat does not inhibit the small cell lung cancer cell growth per se. It inhibits the amoeboid migrations that probably, leaving the tumor site and get to the, so clinically not much value, but very interesting. Oh, I'm going a little bit slow. If I have 10, can I have 10 more minutes? I'm just showing another provocative and ongoing study. I already gave the punchline. TAD stimulates liposomal RNA transcription via satellite 3. Here's a couple of things quite provocative. Liposomal RNA transcription is by pore 1. mRNA is by pore 3. They're using very completely different mechanism. A lot of common transcription basic factors are very different. Second is satellite 3. Satellite DNA is short repetitive DNA sequence, which often existing in human in the heterochromatin silencing. But I'm telling you, said pore 2 transcription activator is able to pore 2 is able to activate pore 1 and the satellite 3 to promote gene expression. So it came from the following. Oh, I forgot to put his name there. Sorry, Matt Franklin, who was a postdoc. He's finishing up that. So when he looked at where's the TAD, he uses EGFP TAD endogenous tag and look at it in the cell. And he found that some cell has 2 very bright dots, sometimes see 1, because this is a confocal section. So TAD forms this kind of punctate staining. And we know this is probably not a so-called phase separation. When we use hexane diol treatment, it is still there. And if we use TAD antibody to stain the nuclear, we also see those punctate. So we want to what they are. And Matt did a chromosome spread. So these are mitotic chromosome in the human MCF10A cell. This is the cell line from the female, so there's 2 Y chromosomes. So the most bright spots is chromosome 9. Then he see, chromosome 15 probably second, 14, 22, 21. That's quite a few those kind of dots. And when we look at what kind of DNA they are, so this becomes interesting. Chromosome 9 has many these human satellite 3 repeats. That's probably more than millions of copies there on chromosome 9. And so these give a perfect match to the dots we see on the chromosome to where the satellite 3 DNA repeats. So suggests there might be on satellite 3. Satellite 3 human repetitive sequence. And so here's the satellite 3 repeat sequence. It's GGAAT, GGAAT. Keep it repeating. And when we input the satellite 3 sequence, and this is the TAD binding sequence, GGAATG. So 1.2 units of repeats give a perfect TAD binding site. And if we use satellite 3, look at what the DNA binding is, the very top are TAD, and there's others. And some of them we are confirmed. They are also indeed other transcription factor potentially can bind to satellite DNA. But this is a bit odd because I said satellite DNA are in heterochromatin. So we want to confirm they indeed binds to satellite 3. So first Matt made a zinc finger. You can create your own protein, zinc finger, to recognize a specific DNA sequence. So he made a zinc finger sequence, recognize satellite 3, and put a GFP there. And now we know these sequences, these protein bind to satellite 3. And then we look at the scarlet fuse the TAD, and they are binding to the same site. So clearly they are binding to the same site. And we did called a DiMello sequence, because satellite 3 is a repeat. You have to do a very long reading. in case there are a few very low frequency changes, so you can map to the chromosome. Actually, when the human genome was finished, the satellite DNA was not mapped there until very recently from telomere T2T, when they assembled the chromosome. We know where the set, but so many repeats, you don't know how many it is. In any case, so when we did that DeBello sequencing, we are able to map indeed the satellite 3 is TAD binds to satellite 3 in the human genome. So the question is, what is the function, right? Why a transcription factor go to a satellite DNA? Now, interestingly, so our genome has about maybe several hundred copies, maybe 200 to 400 copies of ribosomal genes. Ribosomal genes always form tandem, many copies together. And all ribosome gene cluster, RDNA cluster, are flanked by satellite 3 DNA. The opposite is not true. Not all satellite 3 DNA next to ribosome gene. Like these don't have ribosome gene. Those has a ribosome gene. Those has a ribosome gene. So the satellite 3 DNA is next to ribosomal. So we knock down TAD, and we see there are decrease or total RNA to DNA ratio. Now in our cell, right, the majority of RNA, 80% of RNA are ribosomal RNA, which is the vast majority. So next we want to look at is, does satellite 3 really regulate the ribosomal transcription? So first we did the following. We use our this one, the zinc finger protein recognize satellite 3. Now we bring a repressor, artificial repressor, to the satellite 3. So here's the control. The green, we are standing for total RNA synthesis. And in this green are the nucleolus. That's a ribosomal RNA transcription. Essentially, in the nucleus, you don't need to do nothing anything because ribosomal RNA is the vast majority of the transcriptions go to ribosomal RNA. And if we bring the repressor to satellite 3 DNA, we see very dramatic reduction of ribosomal RNA transcription, newly synthesized ribosomal RNA. And this is, different cell lines showing the same thing. So it's able to do that. Secondly, we want to do that. What happens, YAP and TEAD, we first build an artificial reporter. So that reporter is have many satellite 3, 104 repeats in front of a typical rDNA promoter. And then we have a reporter and look at the regulation. And the bottom line is if we overexpress TAD and YAP, and this one, this is the control, increased this POL1 reporter activity. So directly can activate the artificial reporter. If we use zinc finger targeted satellite 3 binding, bring YAP there, also increase that. So it can do that. And finally, we want to ask is, does the endogenous YAP involved in ribosomal transcription? So one way we did that is in this cell, if we knock out the YAP tags, the cell growth is poorly. So we want to do it temporarily in short time. So we use the first one, use the degradation system. In the strep it's the same thing. I just showing the model. So basically, our protein of interest, YAP, is fused to this protein, and this E3 ubiquitin ligase. You put this is a PROTAC, and within 15 minutes, 30 minutes, very nice degradation. And then we can see if we label nucleolar RNA synthesis, once we degrade the YAP, the nuclear RNA synthesis is significantly downregulated. And we can confirm in 3 different independent clones. So with that, we would like to propose the following model. So as I said, in our genome, there's a ribosomal DNA genes, multiple copy all clustered together, flanked by satellite 3. and the TAD is there, and when signal is right, and YAP come bind to TAD, which then turn on the ribosomal gene expression. So, and then we found YAP not only able to turn on POL2, which is all the past study on, is also able to stimulate the POL1, RNA polymerase 1 transcription. YAP is able to interact with some of the basic transcription factor, which recruit POL1. POL1, POL2, that use, many of them are very different. So that's interesting. Second, the Hippo pathway can control ribosomal biogenesis. And another one that we uncovered a function of satellite 3, which is previously essentially nothing has been studied, satellite 3. And actually there's other transcription factor binding to satellite 3. We don't know, we haven't work on that yet. But it's quite interesting. I think it really affect how we're thinking about complete separate paradigm of POL1, POL2 transcription. Some of them maybe can be shared. With that, I would like to acknowledge, I talk about the small cell lung cancer work, which is mainly done by Wu Zhenming, who is currently in my lab, and Matt, who was in my lab at UCSD, and now he's at Stanford, is finishing the satellite 3 experiment. And the TAD inhibitor was done by the company Vivace, Tracy Tan, and we collaboration on the small cell lung cancer with the Li Qingqi's group at Fudan, and Nicholas Ortemos, who is at Stanford, currently Matt is there finishing that project. And I would thank you very much for your attention. Sorry, you had a few minutes.
Thanks, Dr. Wang, for the wonderful talk. This is a great talk for the students to learn the stories behind the paper. So, any questions from the students?
Right, that was really fascinating, and so I'm really enjoyed your talk. And so I got questions. So first is your inhibitor for the histone acetylase. So we know the histone acetylase is like it's like hats. They are kind of universal for a lot of things. So if you use this small molecule to block it to increase the YAP transcription, will you also like affect a lot of other cell activities? Mm-hmm. Yeah, will there be like quite big toxicity in clinical trials?
Yeah, so I'm not an expert on that. So histone deacetylase, like HDAC1, HDAC2, they can form complex with multiple different complex. Even the SYN3 complex, actually SYN3 has AB, or the R-CoR complex, 1, 2, 3, they are the catalytic subunit, probably is the same HDAC. Now, antennal state is a group of families. The benzamidine family histone deacetylase. Before our study, previously, other group has shown it is more selective towards the R-CoR complex, mainly disrupt the complex, R-CoR complex. Now, TSA, traclostatin A, which is essentially inhibit probably all histone deacetylase, very potent. I might be wrong. I think TSS directly inhibits the HDAC